to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 71st episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I've been in the LBA and cell and gene therapy space for over 29 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for around 29 years too.
John and I are both subject matter experts at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas. John is in upstate New York, and our producer is in Missouri, so we're all over the map here today. We are amped up to have you listening to the 71st episode of the podcast, which is now available virtually everywhere. When you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener, to provide some feedback. This podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. So again, we're amped up to have you here, and we are looking forward to a fun episode. To kick us off for podcast number 71, John is going to go over today's podcast topic and a bit before we jump into the news and resources.
Yes, so as usual, we'll spend some time talking about some medical research news, things that are on the horizon and what could be in our futures. And the main topic today is talking about drug development and where bioanalytical fits in. We thought it'd be a good opportunity to really go back and talk about what bioanalysis is. We talk about an awful lot of stuff in the context of this podcast, but we just thought it might be nice to really sort of reset on the bioanalysis piece as we set ourselves up for future podcasts.
So John, you want to kick us off with the latest news and resources?
Sure. Let's whip through some of these. Yeah, the first one: inverse vaccine could help tame autoimmune diseases. And this is some work that's been done, very early work in mice, but it's looking at, well, obviously the problem with it's basically it's looking at diseases like lupus, etc., where autoimmune diseases, where you're basically the antigens in your own body are actually the problem. And so there has been early work done trying to alter, I think it was for mice in experimental autoimmune encephalomyelitis, where they attacks myelin insulation. They attached protein fragments to help insulate those proteins from the immune system, but this is a different approach. It's a group at the University of Chicago where they are looking at targeting self-antigens to the liver, and the way they're approaching doing that is actually to add a chain of sugars to those antigens, which then directs the antigens to the liver. Liver breaks them down, and actually the liver is the organ for sort of determining whether something should be attacked or not, and it's basically fooling the liver into thinking these things should be okay. And so they've done this. They did it with egg protein in mice, saw a strong immune reaction, then did it with sugars in mice. Sorry, attached sugars to the egg white protein, and it didn't trigger the same reaction. And it's sort of early work, but it could be a promise for the future for obviously diseases like, say, like lupus that are actually very debilitating, or multiple sclerosis as well. Sorry, just dropped my microphone, but we're back again.
As long as you didn't drop, John, we're okay.
No, so I passed it over to you.
Yeah, no. Well, this is like the holy grail of there's a lot of holy grails in immunology, John, but the idea of like tolerance and those are still even the article really emphasizes, like, for the last 50 years they've been trying to figure out ways to kind of elicit what breaks tolerance and what causes tolerance. And what they did here was, and they know this, right? You could take egg white proteins and inject it into mice, and they make copious immune responses. However, I think of it as sensitizing. That's the medical word that I've heard used. Yep. When they sensitize them with sugars against or sensitize them with the proteins against the egg white, they somehow they don't generate an immune response, and thereby like kind of break this or become tolerant. So it's a fascinating thing. It just hasn't been done in humans yet.
No,
So
exactly.
it's a major step. It's I can't emphasize enough how, people have been researching this for 50 years, and now we've got it's an advancement in a mouse model, right? This
Yep.
encephalitis model, which is a great autoimmune disease model to really help with patient care. But yeah, pretty cool little study, and I think we could do a whole podcast on how autoimmune diseases, as well as like the concept of tolerance. And then they talk about suppression, which is still the T regulatory, T suppressor cells, all the rage. People study them, but I got to tell you, John, there's still a lot of unknowns about how they are triggered, right? Bridging the innate to adaptive immune response for suppressor cells and regulatory cells is still, it's every day that they're unlocking more doors, but it's still not a completely clear understanding of it, as well as like central memory, which is just making my head hurt a little bit, because how does it establish itself? Because in theory, all the memory cells in your body need some sort of like antigen depot in order for it. So that's like part of where things might be deposited in your lymph nodes. And there's just like this like little bits and pieces of like, from viruses to everything else, allergens and things like that, where they're still kind of in your body, kind of providing the signal to the regulatory cells to keep it so that there's central memory. So there's a lot in there. I think I said, and some of it's, people might disagree with my thoughts on it, but it's a pretty active area. And you've got like two different camps. You've got people in the vaccine space who are trying to prove one hypothesis, and really you've got people in like the transplantation space who are trying to prove a different hypothesis, if that makes sense, right? One is trying to, the transplant, they want to tone down the immune system. They want to make it so that you can tolerate an organ. And then the vaccine people are opposite, right? They want to induce an immune response. They want to make it central.
Yep.
Well, this is a great topic and a really interesting article, and hats off to the.
And actually, yeah, one thing I missed until reading it just now, that Jeffrey Hubble, who's one of the leads on this, actually founded a company, and they conducted a phase 1 trial to establish the safety of this approach in people with celiac disease.
Oh, great!
And so they are moving it forward to see if there's potential for the long term, So yeah, we'll see where this goes, and then I'll move straight on to the next one, unless you get anything else to add.
No.
Because you talk about holy grails, and actually there's been some research in the UK. Pharma Proteomics Project uncovers treasure trove of new leads on drug targets and biomarkers. Looks like we could have a whole load of holy grails resulting from that, and it's basically. Collaboration between 13 pharmaceutical companies and the UK Biobank has uncovered a trove of new leads for drug targets and diagnostic biomarkers, revealing thousands of previously unknown associations with common genetic variants and proteins circulating in the blood. This was the Pharma Proteomics Project, and it basically following on from a lot of work on genetics and disease, pairing existing genetic data to a new analysis of about 2,900 blood proteins from 54,000 people. So I thought that was fascinating in terms of what might be the. It's like we're happy to take on people's projects, but if they've diagnosed or identified a whole load of new targets and biomarkers, hopefully downstream that's plenty of work for us in terms of what we can expect.
Yeah, so much to talk about with this one. First, I think it's the collaboration, right, John? It's great to see that kind of maybe building a little bit off the COVID collaboration that you saw out there, and to see that there's 13 companies and academic institutions all coming together. That's something to be proud of as a community, to see these pharmaceuticals collaborating with the UK Biobank. There was some discussion about that at AAPS, and this is the way to really advance medicine. It's got to be more sharing. So I think that's the first kind of incredibly powerful thing about this, incredible item about this particular article. The other is 14,000 genetic variants leading to 54. I mean, it's unbelievable the amount of data they said. In fact, a couple real famous people in here talk about it being like the they say it's it's a great hypothesis generator, right?
Sure. Yeah, absolutely. Yeah.
which is cool, right? I often say, oh, what's our hypothesis? So a great hypothesis generator is in and of itself like a major stimulator for the community. And then it's fascinating, this protein called GDF15. And that one really caught my mark because it could tell you a little bit about. They talk about it being able to predict a person's age, sex, BMI, liver and kidney function. That's powerful, John.
Just as a
I don't even know
predictor.
I don't know if I want to know mine.
Yes. Yes.
Yeah. No, I don't know if you have any thoughts, but
No,
it's just a lot.
So basically, but just a bit of background. The biobank worked with a Swedish proteomics company, Olink, to test the blood samples. And then the actual companies involved are Alnilam, Amgen, AstraZeneca, Biogen, Bristol Myers Squibb, Calico, Genentech, GSK, Johnson & Johnson, Novo Nordisk, Pfizer, Regeneron, and Takeda. So yeah, I mean, like you say, it's great to see that collaboration. Pulling this together, because this is, like I said, this is the sort of thing that can really help us in the long term.
Yeah, that Olink technology is interesting. It's something we've evaluated. We may pull the trigger on that sooner than later, now that I see it's used here. But that is to data, it's still semi-quantitative, John.
Sure.
So,
yeah.
But still, it doesn't. It's one of those things where you grab all this incredible data and it's like, okay, what's the next step with it? I think
sure.
the article one author kind of is quoted as saying that. It's like, wow, this is so much to take in, and I think it says kind of really using it as some sort of predictive way to help predict, kind of help you with your health, right? Longevity of life. Yeah.
But I think it helps if you can identify the proteins are there, or you've got this whole new set of data on the proteins that are there. It helps build understanding of how things interact, and there's obviously a lot more analysis of the data to come on that to really then look into drug targets, and then what can be done to address those conditions, and develop drugs from there. So I mean, it's just this foundational piece that is obviously could drive a lot of stuff in the future.
Yeah, it's called a huge heroic effort. I couldn't agree more.
Exactly. Yeah.
Still got to put the hard hat on now and translate it, right? That's what comes next, like you said, drug targets and stuff like that. John, maybe we move on to the next one.
Sure. Yep. Yeah, and this is interesting because we focus a lot on large molecules and gene therapies, cell therapies, etc. But small molecules have been a large driver to where we are today. And this was actually an article in, I think, it was in Chemical and Engineering News, why small molecule drug discovery is having a moment. It's a long article. It's worth a read. It's really saying that it's obviously talk about how historically small molecules have been developed to attack to basically hit a target, try and disrupt that mechanism and go at things that way. But actually, small molecules are increasingly being deployed in different ways to help address unmet needs, etc. But also the mechanisms where people are looking to identify small molecule therapies is also changing. So there's a couple of things come together there that really small molecules are not. I remember sitting in meetings years ago, and people at the company I was at the time saying, "No, small molecules are going to be over soon. There's no future in them. We're only really interested in large molecule." And here we are. I mean, we know that a substantial part of what we do is small molecule, particularly on the LCMS side. But there's because the field is moving so quickly and people are thinking about what molecules can do, then small molecules are having a renaissance, and it's not just about designing the drug to address a target. It's actually looking at the molecules and how can they help make other things happen, and we can touch on that. But I at least want to throw that out as a introduction.
Yeah, John. I mean, I think what you said, we all thought maybe 10 years ago, it's like, oh, the large molecules are just going to dwarf small molecules. And we've talked about that on this podcast here over the last few years, but it's still far more small molecules approved. Top 10 selling drugs, I think 9 out of 10 are still, it's like 90% of all drugs sold are still small molecules. It's off the charts here, right?
Yep.
Yeah, on a global scale, around 90% of all drugs sold are small molecules.
Yep.
And that doesn't really surprise me, given I think the cost of biologics, and even like the ready, like, think about what small molecules are treating, things like cardiac and more where biologics is more in that immuno-oncology space. But anyhow, it's a really it is a very long article, John, and it has a lot of detail around the why. Like, why are small molecules still so they touch on, like, a lot of small molecules that 20 years ago chemists thought were not druggable. That's a word that small molecule people use. I've had debates about the word druggable, but it's kind of a funky word, but you know the word well. And it really has to do with what they say something about covalent inhibitors and induced proximity molecules. So maybe you want to touch on that a little bit, but I think that's the advancement. Like, it's not like chemists have sat around idly throwing their, kind of in the fetal position with their thumb in their mouths saying, oh, there's nothing we can do with small molecule. No, they've continued to build on and I feel like small molecules been around 50, 60 years now. And so they've just built upon it. And things that were, so when they say druggable, meaning it was too toxic or, right, and they found ways to make these small molecules into drugs that you can give to people. Is that about right?
Well, some of it is definitely that. I mean, yeah, that becomes ADCs, where you can actually, can give things that are toxic to address cancers. I mean, I think there's a solubility piece as well.
Oh, that's it. The druggability. So it's the solubility
and bioavailability, things like that. Yeah, I mean, I think they all play into that.
Well, that's why we have Captisol, right? Captisol's our big friend in this space.
Well, yeah, exactly. I mean, it's and of course you've got the lipid nanoparticles for bigger molecules, but but that does help. I mean, this article touched on that small molecule isn't just the traditional less than a molecular weight of 500. They get bigger, and that's where lipid nanoparticles are coming in terms of helping deliver those as well. Where you get into the low thousands, which we still consider as relatively small.
Yeah, no, and there's some talks around how chemists do some post-translational modifications, such as deubiquitination.
But that's again that's all like using the molecule to help drive the proteins. It's like you say about these protacs, the proteolysis targeting chimeras, which they have binders at both ends which draw together two proteins in the cell, add ubiquitin tags to flag the protein of interest for degradation. And there's variations on that. There's regulated induced proximity target chimeras, which hold on to stabilize and inhibit problem proteins, and lysozyme target chimeras, LYTACs, which specifically mark extracellular proteins for destruction. So things like that. I mean, we also know of other, again, going back to ADCs, there's so many different mechanisms that people are looking at, but there's also there's where you add the ADC and add a subsequent small molecule to actually activate the ADC once it's in situ. So there's things that there's another small molecule that's doing a job, but it's not the actual drug. It's actually helping the mechanism of action of the therapeutic.
Yeah, well, that bioanalytically creates a whole bunch of methods and uncharted territory a little bit, but it's a really fascinating read here, John. And I see that they even quote the Lipinski rules, which for some reason I kind of know that a little bit from. So this is, I can't tell if there's some impact on changing those rules, right? Or are those still?
It's just, I think it's just saying that the rules, for people who don't know, it's based on successful drug candidates were often smaller than 500 grams per mole in mass and lipophilic. And I'm not sure what the other 5 rules are, to be honest. But a lot of these drugs are outside the 5 rules that Lipinski outlined, because his rule was really, it's like a successful drug to basically address a target, whereas, like I say, a lot of the roles that people are looking at here are to do different things, or to help either a small molecule or help a large molecule address, the target of interest.
Yeah, and then there's all sorts of like technology advancements.
Yeah,
And think about AI. I wonder the impact of AI, and maybe that's something we'll talk about on a future
we have touched
podcast.
on that in the past. I mean, yes, AI. I mean, it's sort of, I remember when people were doing, combinatorial chemistry. This would be back in, I suppose the late 80s, and they'd have labs just churning out lots of small molecules. I think AI is trying to be a more intelligent approach to doing that. So you're not just cranking out things at random, but it's like much more the target, what your ideal molecule might have, and then coming up with variations on that to address a particular target, I would suspect.
Yeah, and I think you touched on the ADC component. That's only going to continue to drive the small molecule development or research. One last question for you here. They talk about um, long. What do they describe it as? Long small molecules or something? What do they mean by that, John?
I think they're just saying it's not. Again, it's something that steps outside the Lipinski rule. So
Okay.
if they're long, they're going to be. It's going to be a much higher molecular weight than 500. So it's yeah. We'd still look at them as an analyst and say that's a small molecule, but it's not a classic small molecule. So it's that's really what that's referring to. So,
Okay,
I
great.
mean, it's one of those things. It's like, it's encouraging chemists to think outside the norm, and obviously companies are, but it's, your small molecules aren't just. There's so much more to them than we've traditionally been thinking.
Yeah, I think the take home is they continue to not they continue to be part of the forefront of drug development, and they're not going anywhere. I think we talked
Exactly.
about that.
Yes,
Yeah.
exactly. That is it, and that's why I wanted to mention it, because we don't talk much about small molecules, but that was an interesting take on it all.
Yeah, yeah.
And obviously Protax and things like that, there will be more discussion of them in the future. It's just that they're still at that stage where they're investigative and none have been approved, but we know there's a lot of attention in that.
Yeah,
Area.
body by chemistry, John. We're getting to that age, so that's a
Sure.
good thing.
Yeah.
Yeah. We got a couple more last articles here, right? And
Yeah,
then-
this is I think this is interesting because it was a couple of news articles came out in very quick succession. The issue here is, obviously we have a huge waiting list of people who are who could use organ transplants, but the issue is that we don't have the supply to meet the demand. So there has been, there's been ongoing research in terms of, can we transplant, for example, pig organs into human? And there's been a hospital in Maryland has done a couple of heart transplants for human patients, people who were very, very sick, and it was like their last chance. And so it was like to help give them a new lease of life. And I think the two patients did benefit from it, but sadly both, in the end, they're looking into the causes as to why they passed away. The challenges here are, potential, going back to our the first article we talked about, is that rejection by the host body of something that's alien to it. So that's like the downside. At the moment, it's been attempted in human. And that group in Maryland have made tweaks to what they do to the organs to hopefully make them more acceptable to the human host. But again, like I said, they're starting with very sick people, so I mean that might even play into things here. But
Yeah.
the other article may actually point to a longer term approach for this, and this was CRISPR edited pig kidney lasts for 2 years in primates. So this was published in Nature. It's a proof of concept study. It's a company called eGenesis have done this, and basically, so they looked at so the xenotransplantation is the transplant of an organ from one species to another, and so this, like I say, this was pig kidneys transplanted into monkeys. and I'm just trying to find. So I think they changed the kidneys with CRISPR in a quite a large number of spots, but they've found that, well, at least in the current format, the
69, yeah, 69 different genes they introduced.
Exactly, yeah.
CRISPR, yeah.
But at least with taking that approach, the organs. I mean, they put them in kidneys from the pigs with transgenes and knockouts into 7 primates. The grafts survived for a median of 174 days. Even meanwhile, transplant recipients who received kidneys from pigs with just the knockouts, and I should say what the knockouts, that's knocking out. So they did 3 edits. One was knocking out genes associated with short-term transplant rejection, adding and overexpressing human genes that regulate important rejection pathways, and then activation genes associated with porcine endogenous retrovirus. So these were the changes they made, and so the transplant recipients who only had the knockout, the transplants who received kidneys from pigs with just the knockouts, 6 of them total, lasted for a median of 24 days. So doing the knockouts and the transgenes, it took them out to 174 days, and the monkeys were all on an immunosuppression gene to obviously help all this, and that's what you would typically do with organ transplants anyway. But one of the transplant recipients who had the full spectrum of gene edits survived for 758 days.
That's amazing, right?
Yes.
That's unbelievable. So, I mean, this is something that, like, I'm bringing me back to when I was getting my PhD in immunology, John. We had all sorts of exams around, well, not all sorts, but one of the classes I was in, we had an exam specifically geared toward transplantation. And really a lot of it has to do with, even back then they were trying to, I went to Pittsburgh. They were the first university to do some xenotransplantation from non-human primate livers and lungs into humans. And they got people to, kind of live a few weeks to months at a time. But I don't think they ever went the 700 days like this. But, you flash forward to today, people are still researching these things. And similar to 50 years of trying to figure out how to break drug tolerance, here we are a good 20 years looking at how to unlock transplantation for xenografts. And a lot of it has to do with the innate immune system and the redundancy of it. And that's why even knocking out 69 genes and all the things they're doing, it just has so much overlap with the way we're designed, so that no opportunistic bug or anything getting into you kills you.
Sure.
So that's the whole premise of why it's so difficult to make xenotransplantation work. And there's like decoy antigen factors is one in particular. It's called DAF. And then all your complement proteins are part of it because they litter all the epithelial of your cells so that, if something, there's an innate immune cell hits that DAF receptor and moves away, well, if it's infected, it goes down, and that triggers the innate immune system. And there's all sorts of things that happen. So when you transplant xenograft-wise, the innate immune system is really the trigger for why it leads to rejection, right?
Sure. Yeah.
And here we are looking at what's the latest and greatest technology out there. CRISPR, it's an incredibly powerful tool. It's really getting my heart pumping thinking about the day when, I might need an organ, and I think I can just dial one up from maybe a pig or even a cultured one. That's even the wave of the future, John. Right? That's something we can be able to do. Yeah. If we want to go real Star Trek wise in terms of the advancement in medicine, but it's an incredibly powerful way to tackle this issue with CRISPR. And I can't emphasize that enough. That this is some of the CRISPR technologies have some, I'll say, safety concerns that are things that kind of I still wonder about. And they're making advancements. They're modulating the CRISPRs, and there's some articles I saw that, and maybe we can cover that a little bit more about what the latest. What is the status of CRISPR, right? Because CAR Ts now there's allogenic versions of them, there's off-the-shelf versions of them, there's CAR B cells, CAR macrophages. I mean, that space is exploding. And a lot of the safety issues, there's still occasionally one here and there, but they've overcome it by, engineering it the right way.
Yeah, sure. Absolutely.
And I think this will be a major advancement for CRISPR. Yeah. It's a huge advancement to be able to take things and learn from animal models and even, periodically go into the human with it in these xenotransplantations. But really cool article. And stay tuned. Although I would have thought the the mechanical heart would have been the wave of the future. The Jarvik, remember that one?
Sure, yeah, I think for the, like I said, for the patients in Maryland, they really had they had no other option, you know. That was certainly the gist of the article about the second patient.
Oh, I see. So
And
sometimes they exhaust all means.
oh, absolutely,
Yeah,
yeah.
I got you. I got
I don't know. They didn't go into what they tried, but honestly,
you.
he was a sadly he was a very 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 sick person, and it was like something that he said, if I can get, you know, a couple of extra months, and then let's do it type of thing, you know,
Well,
because
700 days is a couple years, right?
yeah, that's on the monkeys, but the human
Oh, got
I mean, I would have think. I mean, to me, it's, I would have thought. Obviously, I'm not a transplant person, but I would have thought that the heart has got to be a much more complex organ to do like this compared to something like the kidneys, you know. So
Surprisingly not, though. But yeah, you would think so. I don't know if that's a true. I guess I don't know. But yeah.
I don't either. I'm just I don't know. I don't know. It just sort of.
Well, we don't have a mechanical kidney, and we got a. You can get a mechanical heart, but I want to look into that one myself in my spare time because I owe so much of that, John.
Sure.
But no, great article there, John. And I know we got a pretty dense topic here, one that's near and dear to our heart. Do you any closing remarks on the news?
No, I think let's move on to the main topic.
Yeah,
it makes sense. I'll give a quick introduction. It's, you know, we've been doing this. We're now up to number 71 on this podcast, and we jump around all over the place. And we thought, well, actually, it might be good just to touch back at the beginning and talk about drug development and where bioanalysis fits in. Why are we talking about this? You know, once a month or once every few weeks. Why do we have a podcast talking about bioanalysis? What's the driver for this? And so that really is our topic. It's, you know, it's like Dominic says, it's near and dear to our heart. So yeah, we hope we don't get too carried away talking about bioanalysis, etc. But it was just, I thought we thought it made sense to just do more of an introductory session to set us up for the next 70 podcasts. You know?
And I think we had a question say, hey, can you guys go over that's spawning some of this too, John? But continue. It's an audience request, is what this is.
So that really is it. So I mean, I think if you want to get things going, let's get talking.
Yeah, I think, John, like just looking at your agenda here, it's an initial drug design and discovery, right? And for us, when we think, or at least for me, when I think of really where bioanalysis picks up, it's in between that point where, you know, people are doing high throughput screening of given drugs, and that can be small molecules or large molecules. That's not really bioanalysis at that point. They're just, I think that's still more, is it stable, right? Some of the things you talked about before, can we make it? Is it soluble? All that. But as you get into that lead candidate area where you've started to narrow down to maybe a handful or less, that's where we pick up, right, John? Analytical method. Go ahead. You can jump in.
I'll jump in there. Yeah, I think for me, it's what you. Once it moves into biological fluid, then or biological system, but that might be, I mean, mainly it'd be like into animals, etc. as well, considered, but there might be some additional things outside of that.
Yeah, we can do a little upstream, right? We can do some in vitro studies.
Yeah, exactly, exactly. So we I mean, I think this was more pertinent to where I was before, but certainly the early stages where you're looking for small molecule, particularly, solubility. They do initial P450 work, so in terms of a screen, in terms of potential and metabolism, etc. That all plays into the early compound screening. We don't touch on that. There are labs who are specialised doing that, and they crank through that stuff. And certainly, my colleagues in the past, they had an automated system to do everything, and the vast majority of drugs, those set methods would work, and then that sort of moves forward to identify those of potential to go into early discovery. And these might be, you may have an early discovery might have a number of similar structures, similar drugs, all with structural variations, and you're just doing a comparison in animals to see which ones behave best in terms of the pharmacokinetics, which for us is how long does it hang around in the body and how high a concentration does it reach in that time. And so you're getting a measure of how the molecule, how long it's there before it gets eliminated. Because for some indications you actually don't want it to hang around long. Other indications you want it to be there for a longer time and much more. The half-life is important of the drug in terms of what you're trying to use it for.
Yeah. So John, you nailed it, right? Like there's the early phases here are when you start to want to put things in a matrix. And I think to take a small little step back a little bit, it's also that manufacturing component of things too, John, right? Like people have some access. You've got to be able to make small lots of research grade, really not anywhere near a GMP grade of your drug. You've got small quantities that you can produce. Mind you, you've already done some of the screening where now you got the lead candidates. Those lead candidates, you're going to make some small batches of those, right, John?
Yep.
And then you're going to do some of that in vitro safety tox studies that really isn't our sweet spot. We can do it, but I'd say it's, we're a little more upstream of that, and it's just not our, it's not our primary business line. So there are other CROs that can do that. But once you start to get quantities and you want to put it into an animal and you need a rough and ready method, that's really where we pick things up. And we can do some dose form analysis on it, which is more of an analytical method. And but primarily when the earliest stages in bioanalysis that, bioanalytical CROs can do, especially us here at KCAS, is when you've got lead candidates, a couple of them, and you want a rough and ready method that you've, probably given doses to some mice, and you just need, as John described, some sort of PK, not looking at efficacy at all. It's all about the PK and in terms of its half-life and Cmax. And that follows the biological pathways too, and we can maybe that kind of leads into our second topic. But before we go into that PK and the ADA component of it, because that becomes somewhat, we can talk. I'll touch on that in terms of where we look at that in like a non-regulated discovery space. But John, any other thoughts on that initial drug?
Yeah, I think I want to talk about the bioanalysis. So obviously, I'm on the LCMS side of things. For LCMS, if you've got drug in a biological fluid, you want to get the drug out. You then want to separate what comes out, what you've extracted from other components that will have co-extracted, and then then the mass spectrometer is what we're using as a detector. So we have 3 steps of optimization. So we have the extraction, chromatography, and mass spec piece. When we're doing these kind of projects, the emphasis is on speed. So it's speed, and we will be missing some of the things that we come to rely on later on in the development process. So we internal standards that we use to normalize our data. You won't have an optimal internal standard, but again, because everything's relative, it may not matter too much. You are probably, clients are going to try and push you to go for as generic an approach as possible. So it's protein precipitation, so it's limited cleanup. They may go rapid chromatography, or again, you may have a 1 or 2 chromatographic approaches, and that's it. You maybe switch between 2 different column setups, and then the mass spec is you've obviously got to tune it per compound, but that would probably be using the tuning of the instrument rather than really doing much optimization of your own. So this is like a speed piece. As we move forward, then we get more sophisticated with our each of those steps, the extraction, chromatography, and mass spectrometry. But then I'll pass it back to you.
Yeah, I think there's a lot to unpack what you said there, and I want to reinforce it because I think it's independent of the drug type. In these we'll call it discovery setting, John, the methods are that rough and ready as you described them. They are not methods that are going to translate into GLP. They're generic methods. They're really just giving you that, as you said, speed. We're less worried about just, if less worried about the actual like sensitivity of the method, right? It's more around, hey, I've given a bolus of this. Can I see it? Does my PK, I'm taking a sample oh yeah, my PK profile looks good. it's the same way in the biologic space, but it's specific to mAbs, right? If it's not a monoclonal antibody, then there is a different kind of discovery pathway that has to be taken. But if you're doing large molecules or biologics specifically that are monoclonal antibodies, then we take a very similar approach with the bioanalytical methods.
Yep. And I do think as the molecules become more complex, then it's more difficult to apply that generic approach in LCMS. So, when you go and you start talking oligos and proteins and things like that, we can certainly do them by LCMS, but it isn't just take an assay off the shelf and run them. There's going to be more background.
Yeah.
Even for these early stages, you're still going to have to spend more time getting an assay that is a bit more robust, just due to the nature of the analyte.
And what are we really talking about? It's the building blocks of the method, right? Like for you, it's a standard, a deuterated internal. All that is those are reagents you have to like generate, custom reagents. But surprisingly, John, you can get away with, I'd say, even in the biologic space, even if it's not a monoclonal antibody, there are so many antibodies available. Typically, there's got to be some commercially available source that is suitable for a discovery grade or a research grade method, or on the mAb side, it's generic methods. I don't want to, and so that's kind of the concept here, because you're still trying to identify your lead candidates, and you don't want to spend 3 months and $30,000 trying to make some sort of antibody against that biologic. If it's you'd have to make many of them to be specific to it. So I think the industry as a whole is a good enough approach, a rough and ready approach to help you narrow down things. So I think that's important to emphasize, that most of the methods that are used for research grade testing, generic methods, they don't really have that application into a true regulated method. I want to emphasize that. But on the plus side, you can usually historically get away with using commercially available reagents.
Yeah, and that's when.
And then I'll just on the biologic side, periodically, those methods, if it's the right configuration of the method, it can hold true into some GLP tox in murine and perhaps non-human primates. But for sure, you need to completely overhaul the method when you go into the clinic.
Yeah, and just as a complementary piece, when you talk about generic mAb assays, we can do that by LCMS, obviously. Rather than Dawn and their team have done a lot, but because they don't need a specific antibody, they just a general antibody that will pick things up is great, because they're looking at the LCMSs, doing the heavy lift.
John, you're right. I'll give us a shameless plug. That's what makes us the greatest bioanalytical CRO on the planet. We have the ability to do generic methods for small molecules, generic methods for large molecules, and we have multiple platforms to do the large molecule component, because that mass spec by hybrid mass spec for generic methods are incredibly sensitive. If you have bad antibodies that aren't working in the LBA space, often they can be applied in the hybrid LCMS space, because we've got a really robust set of peptides within the framework of a human antibody.
Exactly.
Yes, so it's an important. Thanks for bringing that up, and that is really a nice. When you are looking for discovery programs, that's the flexibility and nimbleness we can provide for you at KCAS.
You're right. It's knowing the universal peptides that we can then look at per antibody that are across the board. That's the key to success, and they can develop assays very, very quickly to do that.
For antibodies. So I'll just touch on the ADA component in the regulatory, in the this discovery space. Most people actually don't invest much into it. However, there are generic ADA methods that can be applied. If some larger companies, they're going to do it. But most of the smaller biotechs, they're not doing any sort of true immunogenicity, and the regulations are certainly favoring towards investing less in a non-clinical setting and pouring your resources into a clinical setting. So there are some groups that will still have it's a really rough generic assay where you just put the antigen down, you see some signal. If you can dilute the signal, that's good enough. It's a really rough methodology, and we routinely do this. And then it can, as we advance in stages, there's some regulatory leniencies around having like a qualified method that's only a couple tiers and not all 3 tiers, which I'll touch on in future discussions. But maybe we'll stick to our agenda a little bit more. John, I don't know if you have any more on that second, like PK and ADA components.
No, and we probably touched the drug discovery bioanalytical needs already, so we can move into the GLP bioanalysis.
Yeah, we sure did. We really beat that to a sort of pulp with the drug discovery piece. But I think it's important because that does, that really, as you get into drug discovery, bioanalytical needs, I think before we jump into the GLP bioanalysis, there's that non-GLP, non-tox, right? There's that gap sometimes that happens. And or do you consider that to be the discovery space still, John? That early.
I think I because it's non-GLP, I always think of it as discovery, but it's not. It's not really discovery. Discovery is a term that everyone uses as a catch-all, but really it's non-GLP. It's just studies that people are still they're figuring out what's going on with their drug, and it's data that is interesting to them that is highly unlikely to be submitted to an agency, but it helps give them more of a handle on what the drug's doing before going into the full tox studies in GLP.
So I think I can't speak to the small molecule, but in the large molecule, people will often, like I said, make small batches. Of drug itself. They'll 3 or 4 different candidates. They'll put it into some mice, a handful of mice, and we'll do a rough and ready kind of methodology and start to predict. That's all to me discovery. When I hear like a non-GLP tox study, that's usually a single candidate, right, John?
Oh yeah, absolutely. Yes.
That's the differentiator in my mind. When I think of discovery work, discovery bioanalysis is, very different than just discovery research or drug development. But the bioanalysis component is anywhere from 3 to 5 drug candidates that we're doing rough and ready methods on. Then when we move into a true non-regulated or non-GLP tox kind of, that's when people will make an investment in a more robust method that is qualified.
Yeah.
And some people will validate. If you have the building blocks ready and it makes sense, then validate the method to still support because it's ready. But I think it's, again, that's just kind of my thoughts on it. But I think everybody does it a little differently. But I think it's important that as we get into that true like non-GLP to GLP bioanalysis, that's where we're going to dive in a little deeper on.
Yeah, and I think that's for us, that's more where our sweet spot is.
Absolutely.
Well, not necessarily a sweet spot, it's more where we tend to be asked to do work. And in terms of an LC, like again going back to LCMS, you're right, you're going to be going more a method qualification, so you're at least going to assess accuracy and precision. You going back to pure discovery, you might not even look at accuracy and precision because it's all relative. Here you're going to be doing accuracy and precision. You may do some limited stability. You may actually have. You may not have a stable label internal standard, but you have an analog. So again, you're working on tightening up those acceptance criteria. So your assay is becoming more robust, and so there is a progression there. And this is, I think, because you're doing accuracy and precision, things like that, you're getting much more understanding of the bioanalytical behavior of your analyte. So it will give you some flags for when you move to GLP and regulated, because that's going to tell you, is carryover an issue with this compound? If it is, you're going to be thrashing out a lot of the issues with it before you go to the full tox studies, things like that. So there is more attention paid to method development. Again, it's not just going to be that generic. It could be protein precipitation, but you may actually have to look more at liquid extraction, solid phase extraction, other things. And so there's more focus on looking at a method that is more defendable to the regulators, even though the intention might not be for it to be a study that's going to the regulators. You can't guarantee that there might not be some sort of piece of data from that study going as part of the whole package. So there is definitely more attention paid to the robustness of the assay.
Yeah, and I think on the biologics, large molecule space, we have this. I don't know if dilemma is the right word, but oftentimes it's people will be focused on the drug product itself, the kind of making a pilot lot, and will be relying heavily on a commercially available reagent. And that may be suitable for your non-GLP. And actually, John, we've got many cases where it can be used in the GLP setting, but it takes you have to do, in order to generate like an anti-id to your monoclonal antibody, or generating polyclonal sera against a given monoclonal antibody, or even your drug, or even trying to develop some sort of drug specific antibody. Those can take a long time, and often people don't start that in enough time to support their non-GLP talk.
Sure. Yeah.
So that's
Yeah.
when the generic method's used. And then, if the method looks GLP validatable, then oftentimes you do sort of move forward. But it's always a little bit of you lose some sensitivity, and it always has the method itself isn't as robust as when you go and generate a specific commercially available reagent. And that has a lot of upside in not just large molecules, but small molecules as well, because then you can control let's call it the life cycle of it, John, right? That becomes an really important part of it as you go into GLP. So we've got this, some people choose to do a non-GLP tox study, right? That's leading into the GLP tox. And if you make the investment early and often, then you can build a method that is suitable for in fact, if you do it the right way, it can translate from your non-clinical into clinical if you really make the right investment in the right reagents early on. But that's a big investment. You're hedging your bets. What happens if you have to pivot from candidate number 1 to candidate number 2? Now I made all this. What am I going to make an investment in both of them? So I see the dilemma that I kind of created there. However, as you get a single candidate, even if commercially available reagents in the biologic space are looking good, you still should invest in commercially available reagents, because it's going to make for a much more, you should invest in your own reagents, drug-specific reagents, because that will make for a much better bioanalytical method, and that meaning you'll get smoother sailing through the bioanalytical, like let's call it navigating the bioanalytical waters. And then also you get to control the lots themselves rather than relying on commercial sources. So I think those and even, if you rely on a commercial source, sure, you can get enough to put you through a couple different, say, non-clinical trials, but eventually you're going to have to ask that commercial entity to make a new lot, and that's a hold your breath moment. Although oftentimes you can deal with it no matter what happens. But if you own that drug, then you can make enough to have it go throughout the clinical study.
Sure, well. Absolutely, yeah.
So where does that leave us, John? I
think. I suppose we could talk moving into GLP, because again, so GLP, really, these are the critical tox studies that are going to be part of packages to the FDA. It'll be could be a rodent species and then a larger species. Here's where we do want the robust assays, so that the method development for us, ideally you want a stable label internal standard, which is just a heavier isotope labeled version of your drug that you're administering. And basically the reason for that is any variation in conditions during extraction, chromatography or mass spectrometry, the stable label internal standard should react the same to those changes as the analyte itself. So basically you've got, that's the best way to normalize for any variations. When we again, we'll be talking tighter acceptance criteria. So this is when you're at the 15% plus or minus across the curve accepted LQ, where it's 20% acceptance. Obviously you run full sets of QCs. Here's where we do a full validation, which is 3 precision accuracy. You're assessing benchtop stability, freeze-thaw stability, long-term stability, matrix effects, and you'll be looking in human, particularly at hemolysis, and I should look at hemolysis in animals too, but you'll also be looking at lipemic in human. So you're building a much more comprehensive package. about your assay to say, here's why this assay is suitable to support the studies that are obviously critical to the drug moving forward. When it comes to the actual sample analysis, the one thing again that we'll do in GLP versus non-GLP is incurred sample reanalysis, and this is reanalyzing samples, showing that if you analyze them a second time, you get the same results as you did the first time, and we have acceptance criteria built all around that. So there's a lot more checks and balances built into your assay to support GLP studies and beyond.
Yeah, for GLP, it's 6 A&P runs. I'm not sure if you had said 3, but yeah.
We said 3. It's 3 in LCMS, but it's different technologies.
Really? I guess I'm learning something here too. I did not realize.
It's 3 should be plenty. I had other things that I'd also mean there should be selectivity, dilution, linearity.
Yeah.
There's a bunch of there's a lot of set experiments, and that's where we're talking adhering to SOPs, and that's driving everything we do at this point.
Yeah, I mean, on the this is where we can dig in really deep into the weeds on the regulatory landscape. You mentioned selectivity. That's one that has evolved quite a bit in my time here at KCAS. Meaning, before you could do your selectivity at a high and low concentration, and you would be able to do the low spike, maybe twofold above the LLQ. Well, now it needs to be done at the LLQ. That's a big change, and it makes that you see a lot of selectivity issues around that. Sometimes you may have to raise the lower limit of quantitation on a method due to some issues with selectivity failing at the low end, and then surprisingly in the biologic space it can fail at the high end too, because you hit some sort of prozone effect, or maybe saturating the absorbance can cause some issues, or saturating the signal at the high end can cause some issues with the ULOQ as well.
Yeah, and yes, and we have similar issues. We can we have a linear range on our calibration curve, but we can saturate the detector on the mass spec and it starts to go quadratic, and the regs have pushed us much more to go for linear fits, so you will truncate curves on that basis. But one of the other issues that we have,
and the way we get around to LBA is we anchor it, John. Slightly.
Yeah, sure.
Yeah, go ahead.
Just one of the other things that we have in LCMS that you obviously don't have to worry about is carryover, and that's the contribution of signal from one sample to the other. And this becomes mostly a factor for high concentration samples that will be residual in the system, and it's compound specific, but then you'll have a contribution to low analyte sample, and that may truncate your curve range as well. If you've got a high carryover assay, you might not be able to go above a certain amount because the carryover to your low concentration sample is unacceptable. So
and we have an MRD issue, right? Minimum required dilution, that you guys don't really worry about that so much, because the extraction step you iron that out with how you extract from it. Yeah, so this is a great discussion, and I wanted to one up my fish is bigger than yours moment there, John. In case you're wondering.
I'm not trying to be competitive. I'm
me. I'm going to get competitive, no matter what I do. So it's good to. I feel like this is a dry podcast, but so I thought I'd throw some fun in it. But actually, this has been a wonderful dive into the bioanalytical realm of things, and sometimes we take it for granted that our audience always knows this. But I do think we can continue down those performance characteristics, John, because then we do kind of converge again, right? Like benchtop, freeze thaw, room temp. Which, by the way, I found out that I was happening to look at something. CLIA labs don't really do a lot of benchtop stability, which I found that interesting. Where that's a really important piece of bioanalysis, isn't it? The benchtop piece.
I mean, but its thing is, like, for small molecule, people talk about LCMS as being a routine technology, but well, it is, but it also really isn't. You've got to be very alert to what's going on, because there's so many. Obviously, stability is a big part of it, but it's not just in the matrix, but it can be downstream in the reconstitution solution where you're getting ready to inject onto the LCMS. I worked on a case years ago where it was a atorvastatin and its metabolites. But we weren't measuring all the metabolites, and what we were finding was the ISR was failing on the 2-hydroxy metabolites. There's actually a whole series of lactone metabolites too that we weren't looking at. but and we had two instruments running it. One instrument was passing, the other one wasn't. And basically, it was although both autosamplers were chilled, one of them was in a warmer part of the lab, and so the lactone metabolites were actually degrading to the hydroxy metabolites sitting in storage in reconstitution solution, and so they were going high with time. And so there's just so many facets to be aware of,
I could tell you a few stories about shakers having impacts on MSD assays. So very true, John. When an ISR fails, and mercifully it doesn't happen too often, but when it does, those are the little subtleties that you can start to discover. And that's why they're in place, John, right? To demonstrate robustness, right?
Absolutely.
And maybe I think to most of the audience, they know what an ISR is. It's incurred sample reanalysis. But why don't you walk us through that, John? Maybe real fast. Why? Because I think some people might be hearing it and think, well, what the heck is that?
I did talk earlier about what it is in terms of the reanalysis to show
comparability. Like the 67%. How do we do it?
Our acceptance is you take a random sample of the samples you've analysed. You hope to have a mix, or you want to have a mix of samples that are around the Cmax and also in the elimination phase. And like I say, you want to show reproducibility between original analysis and a subsequent analysis, because that then shows your assay is robust and reproducible. The acceptance criteria are 67% of those analysed have to be within plus or minus 20% of each other in terms of the original and repeat value. And if you're above the 67%, you're good. I mean, but it isn't. Again, it's not. It's even if you had 68%, you pass. But that's going to alert you as a bioanalytical person. I want to watch this assay in the long term, and it may be. Maybe the one matrix is more. Say one matrix may be more of an issue than another. Obviously there's different levels of enzymes in different species, and so rat particularly if you got esterases, etc. If you've got something may have more of a problem in rat than it does in other species. So it's you want to be watching that kind of thing. It's a really for bioanalytical person, ISR is a really useful tool to give you confidence in your data. That's what it's all about. It's like you're pretty confident about your assay, but the ISR is like is really a good additional test beyond validation, because samples will behave differently may behave differently
than your QC.
Spiked QCs, exactly.
And John, you touched on the desire to have those. It's 10%, right? Unless it's over, I think 1,000 samples, then you cut it down to
it's 10% of your samples analysed, unless we go over 1,000, then it becomes 5% thereafter.
And the other piece, you're right. You want to have it at different portions of the Cmax, and I think of it as just within on the standard curve itself too is important, right?
Yes,
high, absolutely. Mid, and low. But then there's oftentimes what happens, John. All the samples are clustered in the same part of right? They're all dosed either BQLs or AQLs, and then in a certain part of the curve. So then you're stuck with trying to sample from an area that's all kind of narrow, and that's not a big deal. It doesn't give you that robustness that you'd like to see across the curve. But then if you do have, what happens if 80% of your samples are BQL? Then how do you do ISRs, right? Or if they're
we wrote our SOPs around that to say you wouldn't do it. Yeah, so
but these are topics that have been ironed out over the last decade. These are all things that were swirling in terms of issuing 483s because you did something that the regs are changing so often in this space. Although I think it's ironed out with ISRs. Although I do think there's still some. I felt like there was a recent change there. And then John, to round out. So I think ISRs are a great part of the performance of a method. And we touched on selectivity. I don't know anything else. I guess we can wrap up a little bit.
I think two more topics very quickly. Moving into the clinic is something I do want to touch on, and then biomarkers, because that's also another piece of this. Moving to the clinic is really for us. What we may find is we're asked to go to more sensitive assays, and that may be a challenge depending on, what we've already done in tox species. So it might be that we have to, again, you might be become more focus on the extraction to really clean up away from other components. Actually, another thing I haven't touched on is metabolites and their potential impact on analytes, and so that's something you always want to be aware of. And if there's metabolites present, and make sure they're separated and away from the parent in case of degradation in vivo or during the process. And then so the other tool, that because we have a range of mass spectrometers, that as obviously we've been lucky, this is a rapidly developing area, and the mass spec manufacturers have been pretty aligned with the bioanalytical community's needs in terms of sensitivity. So we have a variety of SciEx platforms. Right now we have the 4000, 5000, 6500 and 7500, and each of those iterations has improved the sensitivity of the assay and the ability to improve our detection limits and make our assays more robust. The other nice thing about both the SciEx 6500 and 7500 is they've a wider dynamic range, so more linear curves over a more extended range compared to the earlier models, and that helps us in terms of what we can do for our customers. So of course carryover is still an issue, but it's human can come with its own challenges, compared to the tox studies, although we will have learned a lot during those GLP analyses. It's just like, say, if you've been asked to go to lower levels, then you may have a whole nother set of challenges to deal with.
Yeah, and on the ligand binding assay side, there's typically we just use the mesoscale discovery. That's our most sensitive platform. They continue to make it more sensitive. They got some ultra sensitive ways to do things. Their S-plex is what they call that. But there are moments where we may look at labs who have developed a method and maybe even validated a method and supported non-clinical using an ELISA. And then when they go to the human, that's when we would convert that ELISA method into an MSD method, which we do pretty routinely. And I'd say on average, we can pretty easily improve the sensitivity anywhere from 10 to 100 fold. And of course, the dynamic range is much larger on the mesoscale discovery system. And if you're not aware, ELISA is a traditional plastic plate that can get X amount of a capture antibody or X amount of a antigen for capture on there. A mesoscale discovery, and then on the ELISA side, you're going to use some sort of enzyme for a colorimetric change, and you read on a plate reader through absorbance. A mesoscale discovery system has it's the plate itself is like one big circuit board, and it has corrugated grooves in there, carbon corrugated grooves that can bind much more of the capture antibody and or the antigen of interest. And then you use a Ruthenium tag and take a picture of it, and that just builds a much more sensitive and increased sensitivity, increased specificity, increased dynamic range. Although, at this stage, ELISAs are still pretty routinely used, and
Sure.
we have no problem validating an ELISA method as well. So I think you wanted to mention biomarkers a little bit. I know we dug in a lot on the non-GLP, maybe the non-clinical piece, but the clinical side, at that point, John, on the LBA side, your investment has to be heavy in your reagents. You've got to have a good handle on that.
Yeah, I just want to mention biomarkers. Obviously, there's the challenge that they're endogenous, so we do have some different approaches in mass spec to address those in terms of surrogate matrix, things like that. Plus, it may be that we're not going to be fully validating, but we'll tend to be qualified because they're not a primary endpoint during studies. I just thought it was worth mentioning. The similar thing with tissues, actually. We'll do a lot of tissue work, but because of because for so many assays, plasma is more important. The tissue may just be a qualified assay because they want to see what it's doing, but it doesn't have to be quite full rigor validation.
We could go a whole nother hour on that.
I know. I don't want to say any more than that. I just thought it was worth throwing those out.
No. There's a lot of strategies around if you need to validate or qualify, how you want to tackle it, if you're looking at tissues or CSF or ocular or all those sorts of things. And certainly we have a rich history of supporting those, and we really do have good solutions to those types of I don't know if they're problems, but to those types of drugs that are either dosed in a certain way or are going to be found in a certain matrix that's not easily easily drawn, small limited volumes of it. All of that we have the ability to, as you mentioned, work in a
Sure.
surrogate matrix, and certainly the FDA understands that with their guidelines. So I think we have John been a
I think we should wrap
this
up.
is one of my favorite podcasts in a while, although I like them all. This is fun to kind of take a small step back and just drill into what is bioanalysis, and specifically some of the challenges if you got small molecules versus large molecules. But I think we have a question from our audience, some feedback, and this would be a good time to maybe have Jeremy chime in and let us know what that question was.
Yeah, Dom, actually have a little bit of feedback rather than a question, and they're from a couple of friends of the podcast that I believe both of you will recognize just by their first names. Brian and Adam say, "Hello, Dom and John. We're happy to be part of the extended KCAS podcast family. You two have been inspirational and leave a big pair of shoes for us to fill, but we're having fun, and we are looking forward to having you both as our guests on the new KCAS podcast series, The Conversational Flow."
That's awesome. We wish Brian and Adam well. We look forward to routinely having them on our podcast, and we'd be happy to join their podcast. But I can't promise that I won't swerve into their lane and go with the flow, so to speak, because flow is still near and dear to me.
Yes, I'll let you do all the talking on that one. So, yeah.
Well, that's awesome, Jeremy. I know they've, I believe they did their second one, right? Is that fair to say? They have their second podcast coming out?
That's right. We have the second episode coming up just after this one. There'll be a regular cadence of they will have the middle of the month that we will post regularly, and then we will continue to post this series toward the end of each month. So listeners of the podcast here will be happy to know that now we're going to be posting a minimum of 2 different series regularly.
Well, they got a little ways to catch up, but I wish them well. And
absolutely, yeah.
I feel like Jeremy, I got to step up my game a little bit. I've seen some of the microphones and such that they have, so we'll talk more about my budget for my podcast. Okay.
I'll get right on that.
Yeah. Okay. John, let's close out, right? What do you got going on? I know we got a lot coming up for you. I mean,
yeah, my life over the next 3 weeks is going to be very hectic. As soon as this podcast finishes, I'm going to be loading my car and driving off to Rochester Airport, flying into the UK, spending a few days in Edinburgh and then Fife with my wife's mother and her family. Then next week it is EBF in Barcelona, and we have 2 talks. I'll be doing one on hybrid LCMS. Musin, who heads our Lyon facility, is doing another talk on urea as a small molecule biomarker. And also I've been asked to join a discussion on hybrid LCMS. So behind the scenes, I'm going to be picking Ratha and Don's brains as to what they want me to say on that. Then after EBF, I'm flying back to the UK. For another week there, spending time with friends who we all turned 60 this year, so it's a chance for us to get together and actually celebrate that milestone together. So it means lots of beers and everything else.
old people stories too.
Yeah, probably. Well, I don't know so much about that, but I mean,
I'm going to call you a boomer. No,
we'll see where it goes. And actually, because I'm actually in the middle of that. So that's 2 weekends seeing friends, and in the middle I'm actually going up to Oban and the West Highlands, just to have a bit of time exploring Scotland to places I haven't been since I was a kid. So doing that, and then coming back here. In the meantime, we'll also be doing prep for a webinar with Dawn Duffield talking about small molecule biomarkers that we're doing with SciEx. So that's something also I have to be working on while I'm traveling. So yeah, a ton of stuff going on.
Yeah, and I'm usually the one that's getting yanked around, but you make my life seem pretty simple these days. I do have a webinar coming up on immunogenicity of cell and gene therapies, but that's later in November. I mean, I was more interested in, I was in my deep freeze and I saw that I had a big brisket in there, so I might be making a brisket this weekend on my big green egg, because I've got I think we've got one of the few warm weathers we have left. Of course, I'm always thinking about when I'm making my next batch of marinara sauce, and I've got some dough that I've got from a local pizzeria, so I might make some, pizza. But my son works at a Minsky's Pizza now, so I haven't been making as much pizza as I'd like, but that's okay. So I don't have anything exciting. I mean, Thanksgiving is a couple weeks away. I'll have some family over, but I don't know. I'm looking forward to some, maybe trying to work out a lot over the next few weeks. And of course, I'm always cooking up something special.
Which Minsky's is he at?
He's at the one on College, if you know where that is. It's not near any of our facilities.
Okay. Then
it's in Lenexa. Yeah, it's pretty close to our house.
Okay.
And,
yeah, so I mean, obviously, I'm not cooking for the next few weeks, but I've got, I got various restaurants booked in Edinburgh and Oban and stuff. So I plan to eat some really good seafood over the next few weeks, So
well, I've been experimenting with a drink. It's called a sidecar. It's cognac and Cointreau, and so it's booze with more booze. But sidecar, check it out. That's a neat, good fall drink or winter drink. Yeah. If you know what a sidecar is, that's a good one.
So one of the things for me to do in Oban is go to the Oban distillery and sample their whiskey. So yeah, we got some stuff.
Now, you have a lot more going. I'm sitting there thinking that sounds like a lot of fun, but I don't. I may make some trips for business wise, but it'd be a couple nights here and there. But John, safe travels to you.
Thanks a lot
for sure. Yep. But I don't know. I think that's a wrap from us.
Yep, I think so.
Yeah, been a great episode, John. Safe travels, and we'll look forward to episode number 72.
Yeah, thank you very much. I look forward to it too. Hopefully, I have a lot to talk about when I get back.
I imagine you will. Yeah.